is very important in the field of oncology as well. There is recent evidence for a possible effect in the chemoprevention of colorectal cancer. And here I've included two papers. Perhaps the most relevant, published 4 years ago by Andy Gescher's group on animal models, on the relevant animal models. The data proves that the administration of these complex mixtures and standardized mixtures produces a very important effect on tumor progression in this particular experimental model. This is the continuation of these studies recently published by this group in 2009. Again, Gescher's group with a group of clinicians, and it's a pilot trial where the effect was studied of administering these anthocyanins in a complex form to patients where interesting effects were found in terms of a possible slowing down of the disease progression. So what products were used in these trials? Well, I've shown a map of the world here. The VACCINIUM myrtillus, which is a particular species, grows in these areas. Today it's harvested still, so it's a plant that is not cultivated. These are the various stages of the harvesting process, right down to refrigeration of the fruit. So it's a highly complex process that also causes the price of these products to be quite high. And these are just pictures showing the industrial processing and preparation of the fruit, right down to storage and the freezing process. And there are various problems that I'm not going to get into detail on, but there are problems of contamination with other species. The VACCINIUM myrtillus has to be recognized and distinguished from other species of bilberry that also grow in many of the same areas and which have different compositions. This is the anthocyanins or anthocyanicides that were shown earlier in terms of their molecular structure, thanks to Dr. Marco. And the extract that is produced industrially and standardized is based on the presence of 5 anthocyanin skeletons, anthocyanidin skeletons with glucose replacement, showing the 5 products that you can see on the right-hand side of the slide. In the extract, there is an ID fingerprint for the extract, showing the 15 anthocyanins present in the complex mixture. And so the HPLC can be compared with other HPLCs of products also obtained in different ways. And visually, you can see that there are quite significant differences between them, between some of the compounds. And today, we can do a lot more than that. In fact, we carry out spectroscopic analyses. This is the carbon-13 NMR, showing 3 industrial batches of product, reproducible extracts, and there is an overlap of the spectroscopic profile, showing that not just the known component, the anthocyanin component of the substance's extract is reproducible, but also the part that is not generally titered in the extract. So this is an excellent result. There are other instruments that can be used, FTIR, for example. That's yet another analytical process for a semi-qualitative examination of the product. Again, here we use this technique to distinguish products that would seem to be identical but are not. VNIR, for example, these are industrial batches produced of the extract that's used by Dr. Marco, and these are extracts coming from other processes, other manufacturing processes, or uncontrolled substances. So there are tools to distinguish one complex mixture from another, one compound from another. And we've been able to create a number of highly standardized extracts that could be potential candidates in the field of oncology and are already being explored in preclinical and clinical trials. many in Europe, others in the United States, and as I said, in oncology. I'll just show you some very quick examples, because they enable me to say, in the time that I have left, that standardization is the first yardstick for defining pure products, reproducible extracts, which in many cases can be used to correlate epidemiological data where the foodstuff has been used as a food in a diet and not as a preparation. These products generally have a composition that resembles the food product, like the bilberry extract I showed you before is an extract whose composition is comparable in terms of dosage to the known portion to the food actually consumed. This is curcuma that I'm sure you've heard about, because there's been a great deal of literature on the subject. It's one of the principles contained in the root that you saw before, and there are hundreds of papers in the literature. Curcumin is almost a biochemical panacea, if you will, as Orsini might say. If you look at the literature, curcumin is reported to have an outstanding number of activities. This is a paper from 2009, and it's a molecule whose mechanisms relate to the inflammatory process across the board. And as well, many of these biochemical passages have been described involving curcumin, and in oncology, this review shows the mechanisms via which curcumin could interact with proliferation processes. However, all of the information that you see reported involves product concentrations that are unfeasible, going back to what Orsini said this morning.